Thank you for joining us today for our launch webinar From Sample to Insight, introducing the Teton Atlas for direct in-sample sequencing. We're excited to introduce you to our latest product updates and the expanding capabilities of the VD24 platform. My name is Jens Dorothy. I'm the Director of Product Management at Element Bio, and I will be your moderator today. A few things before I hand it off to our speakers. We'll be answering questions at the end of the webinar. Please feel free to submit questions at any time using the QA section at the bottom of your interface. We'll try to get to as many questions as we can, but if we do run out of time, we'll follow up with you after. The webinar is also being recorded, so if you want to review any of the information presented today, the on-demand version will be available on our website. When the webinar is over, you'll be getting a short 3-question survey. It's your opportunity to let us know what you thought of the webinar and topics you would like to hear about us in the future. With that said, let's get started. Today, you'll hear from our R&D team about how Element's latest innovations are transforming in situ sequencing and enabling new applications. I'd like to introduce you to our speakers. First up, we have Claudia Jameson, Senior Director of Software at Element, who's joined by Danielle Honigford, Senior Scientist in the Biochemistry Group. Without any further ado, let's dive in.
Thanks, Jens. We can all appreciate that biology is incredibly complex and multi-layered. There are different biomolecules, RNAs, proteins, phosphorylated proteins, and these are all acting together in coordination, largely in the functional unit of biology, which is the cell, and then all the cells are operating together in a spatially resolved manner. And they're signaling to each other. They form complex structures like tissue in different tissue microenvironments. And finally, an often underappreciated layer of all of this is that many of these aspects of biology are incredibly dynamic and changing over time. And over the last 20 years, next-generation sequencing has catalyzed an entire ecosystem of multiomics tools to try to understand this complexity, from bulk RNA sequencing and single-cell RNA-seq to proteomics. Often these methods barcode different analytes, whether they be DNA, RNA, or protein, have users perform library preparation, and then read out the different analytes using sequencing on an NGS instrument. While these methods are extremely powerful, a lot of challenges remain, such as long workflows, multiple pieces of capital equipment being required, high costs, and unpaired measurements that come from all these different assays and samples, which introduce additional costs and require stitching together disparate data types. But VD24 really seeks to address these challenges by enabling the co-detection of multiomic readouts, morphology, RNA, protein, phosphorylated proteins, all from the same cells in the same sample in a single assay with no library prep. Teton consumables enable multiple samples to be queried in a single run, enabling experiments that capture the dynamics of biology from time courses to drug titrations and more. Importantly, all of these analytes are captured spatially with Teton. So this really allows researchers to see the location inside of a cell that RNA and proteins are expressed, as well as the complex organization of cells within a sample. And thus, the VD24 really achieves what's long been considered the holy grail in the biological tools space, delivering sample to insight with no library prep. Our goal is to enable a broad set of sample types that can be directly applied to our flow cell consumable, loaded into the instrument, and then with no additional intervention, researchers can measure morphology, RNA, protein, and phosphorylated proteins directly from their samples. We can also appreciate that with our instrument model with the VD24, it will be placed in core facilities and service labs. So we've also thought a lot about the user workflow to ensure that our end users can perform sample optimization experimentation in their own labs, and then simply fix and ship their samples to a site for centralized processing. With that introduction, let's dive a little bit more into both the Teton and the Teton Atlas portfolios. We'll start by looking at Teton. So with Teton, we've offered a set of fixed panels that cover a broad set of applications spanning MAPK, cell cycle and apoptosis pathways, immuno-oncology, and neuroscience. We've also enabled a broad range of sample types, including adherent and suspension cells with supporting segmentation models and sequencing recipes. The Teton assay enables the co-detection of 6 cell paint markers, 350 RNA targets, and up to 138 protein and phosphorylated protein targets. All of these are co-detected in a single run from the same sample in less than 24 hours. And to elaborate a little bit, one of the things we've been working on is enabling more of custom protein for users who maybe aren't satisfied with the 50 fixed proteins that we offer in our panels. So recently we've introduced the ability to allow users to spike in up to 88 of their own primary antibodies. And coming this summer, we're launching a set of curated focus panels where users can mix and match up to 3 sets of 24-plex protein based on their areas of interest. The full list of the protein targets in each of these focus panels will be posted on our website soon. So if we bring this all together, what does this sort of look like on the instrument and how does this all work? Well, our multiomics detection is fueled by avidity-based chemistry, the same chemistry that we use for our next generation sequencing. We have a set of probes for our analytes of interest, whether those be specific RNA molecules or protein molecules. And with our current Teton chemistry, these probes are attached to a unique barcode that we then amplify and sequence. Importantly, we're able to actually get more output per cell by segregating our barcode sequencing over multiple batches of sequential sequencing. A fundamental limitation of any imaging-based optical system is optical crowding, but we can overcome this by sequencing in batches. How we set up these batches is quite flexible on our end, and at the moment we have a fixed allocation of batches for cell paint, protein, and RNA, but our end goal is to give researchers like you the flexibility and freedom to construct your own custom workflows for your application of interest. We'll have a little bit more on that later. If you're interested in learning more about our Teton chemistry and performance, we recently posted a preprint that you can access through the QR code here. And before we talk about direct and sample sequencing, I wanted to highlight one more example of the power of the EVD24 Teton assays. In this experiment, we were interested in dissecting differential responses to first and third generation EGFR tyrosine kinase inhibitors in lung cancer cell lines. So we took our EVD24 Teton flow cell, we cultured multiple NSCLC cell lines with different drug types and incubation times, and because we had so many different wells available, we were able to pursue complex condition maps and cover multiple scientific questions at once. These cells were then activated with EGF and fixed, and then the flow cell prepped for our VD24 Teton run. In this run, we were able to look at 350 plex RNA data, 55 plex protein, including 5 of our own custom protein spike-ins for targets of interest, and 6 cell paints for morphology. Through traditional means, this experiment would have taken months to acquire this amount of data. But by using a VD24, we were able to obtain this expansive data set in just 2 days, including our cell culture time. If you're interested in reading more about the experimental setup and what we found, you can also scan this QR code to see more in this preprint. So now let's dive into Teton Atlas and our direct in-sample sequencing technology. First, let's start by talking about where we can use direct in-sample sequencing and the power that it really unlocks. So up until this point, everything we've shown with Teton leverages a barcoding concept. And today, many technologies also allow for spatial detection of various proteins and RNA using barcoding chemistries. Barcoding chemistries are great, but they do have some downsides in that they require many custom probes, they have long design cycles, they're not species agnostic, and they often fall short when it comes to applications where you would ideally use sequencing, where you're really interested in the sequence of the thing you are looking at. For example, TCR-BCR, CRISPR guides and optical pooled screening applications, and express mutations. So by being able to directly sequence in cells, this would really enable a lot more for our users and for the environment as a whole. So let's take a deeper look. With Teton Atlas, we actually are able to sequence directly in the cells. So this is compatible with any species and any sample. It offers fully automated, library prep-free, load-and-go multi-omic data acquisition. We can do reads of up to 100 plus base pairs for both 3' transcriptome and targeted RNA sequencing, both of which we'll dive a little deeper into later in this presentation. And this technology is compatible with our Teton chemistries for cell paint and protein and phosphorylated protein detection. These methods can be used together in a single run on the same set of samples, generating multi-omic readout with 0 batch effects. So if we layer this onto what we saw earlier, with Teton Atlas, we can also now look at native RNA sequences in addition to the dynamic responses, the co-detected multiomics, and the spatial resolution of all these readouts. Again, to highlight, this is all in one assay with zero library prep. So it's really transformative, and we can't wait to see it in your hands. If we want to see this direct in-sample sequencing in action, here you can see a cell paint marker highlighted in the background, and then the polynese of the direct in-sample sequencing cycling on top of that. So the Teton Atlas high output assay, which I'll elaborate on in a minute, enables the co-detection of up to 6 cell paint markers, 300 cycles of RNA sequencing, and up to 88 protein and phosphorylated proteins. All of these are co-detected in a single run in the same sample in less than 48 hours. So if you want to get your hands on this, what does it kind of look like? What are your options and how do you leverage this? So we have 2 cartridge configurations. Our low output cartridge will support 3-plex cell paint, 100 base pairs of targeted RNA sequencing, and up to 88 proteins being read out, and that will launch in August. Our high output cartridge, which includes additional cell paint markers for human, and up to 5 batches of 3' transcriptome sequencing, or up to 500 targeted genes for counting applications, in addition to the protein, will launch in Q4. That low output cartridge is actually available for pre-order today, so we'll have a slide with a QR code at the end of the presentation where you can get your hands on it. So let's talk a little bit about how users might use these different cartridges and how they might leverage the batches within them to drive tunable output for the experiments that they desire. So in addition to the cell paint and protein, which we've already talked a lot about, we have 2 different types of batches that support RNA sequencing for counting applications and RNA sequencing for other applications. With this other applications or specialized RNA sequencing batch, we allow up to 100 cycles of sequencing data for users to target guide RNA spacers, barcodes, TCR sequences, and the like. And then with our sequencing for counting specific batches, we allow users to leverage up to 5 batches of targeted or 3' transcriptome data for applications like differential expression, pathway analysis, and cell typing. With this sequencing for counting batch type, users are able to generate targeted custom panels for up to 500 genes or to drive discovery with approximately 2,900 unique genes per cell detected with our 3' transcriptome if we were to do 5 batches of HeLa cells. And we'll see more of this data later. So great in concept, but what does it look like in reality to look at some of these applications? So in these videos here, you can see different polynese being read out from guide RNA sequences, from a targeted RNA GAPDH target, and from the 3' whole transcriptome. Below each video, what's really nice is you can actually see the downstream alignment data showing how this sequencing data is being used. Just to point it out again, this data is generated with 0 library prep and with spatial traceability, really making it quite powerful. So if we want to ask, well, how do users actually leverage all of this flexibility? How do they use it on their instrument? And that's where part of the power of this assay comes with our approach to making this fully programmable by our users for their specific use cases. We'll walk through some specific examples in the following slides, but at a high level, in our Element Bio Cloud Designer, users can build their own custom instrument workflows out of our cell paint, protein, and RNA molecules. The cartridge is then configured to allow the user to drop in various modules specific to their custom workflow, and the software on the instrument will leverage that workflow in the cartridge to produce the data of interest, which users can then analyze downstream. So we can take a look at the software suite across this workflow, starting with the Element Bio Cloud Designer. So here we see a demo where the user can select that they're building a Teton Atlas DIS workflow. They provide a name, some basic information, and then they're able to leverage the modules to select their cell paint, their protein. In this case, they're using one of the focus panels and then adding some of their own custom content. And then they'll go ahead and provide information to target a CRISPR optical pooled screen scaffold set, and then provide a manifest of interesting spacer sequences that they're looking at in this pooled screen. And then once the design is complete, they'll get a full list and a set of downloadable instructions around what they need to order and how to prepare for their experiment. So you'll see here they get some primer sequences, they get information about how the cartridge will be assembled, and then they can use this design moving forward. So once that custom workflow is built, when it's used on the instrument, the primary analysis will handle cell segmentation and morphology metric generation. Users can also resegment offline if they would like to after the run. Then it will handle protein assignment and localization, as well as base calling. And if the user is doing an optical pooled screen, we'll also have optional support for spacer cell assignment and localization on the instrument. And that can be used for spacers or for barcodes. So then after the instrument, what does it look like for users? Well, first they'll take cells to stats, and this will generate their FASTQ files for any of their DIS reads, as well as help with any cell processing. And then after that, if they need to, they can do different read alignment or cell assignment workflows and then bring that data back. And then lastly, they'll be able to visualize their data in CytoCanvas. And we're also working on, for the optical pooled screen application, which Daniel will highlight, the ability to generate cell montages of interesting cells. So with that, I'll kind of summarize what we've seen so far. Teton Atlas is multi-omics compatible, easily customizable. It offers tunable sensitivity. It's highly specific, and it's fully automated, again, with the big highlight here of no library prep. So with that, I'm going to pass it to my colleague Daniel to walk us through a bit more of the DIS chemistry and some exciting DIS data.
Thanks, Claudia. My name is Daniel Honigfort, and I'm a scientist working on Teton Atlas. So today I'm going to introduce the fundamentals of our detection chemistry and showcase some of our data. So as Claudia mentioned, we have two different approaches for direct in-sample sequencing, a 3' transcriptome option, and additionally the ability to design custom targeted RNA probes. Our chemistry utilizes a similar single-sided probe design for both approaches. For the 3' transcriptome, we utilize poly-T probes that bind to polyadenylated mRNA. And for targeted sequencing, we use a probe that hybridizes to specific gene or regions of interest. That can include anything from a spacer, a barcode, splice sites, or just any unique gene sequence that you want to use for counting applications. In either case, after hybridization, we perform cDNA extension, which is really the key step. That encodes the actual target RNA sequence into our probe, which is then circularized and amplified with rolling circle amplification. Finally, we read out the transcribed cDNA using our ABC sequencing chemistry. I want to emphasize that the output of this assay is the actual sequencing information of the gene of interest, not counting a barcode. And for anyone thinking that this sounds complicated, I would say, don't worry. Every step that I've shown here on this slide happens on board with no user intervention. And if I can also squeeze in a quick fun fact about our ABC sequencing chemistry, we're actually able to use the same chemistry here that we use with regular sequencing on Aviti. And in fact, it's this high-quality cycling chemistry that has enabled us in some in-house runs to achieve 150 base pair sequencing in cells, which is a cool milestone. So the fact that our probe is a single-sided oligo and not a double-sided padlock makes it customizable for a wide range of targeted applications. So as an example, I'm showing here a probe design for CRISPR guide RNA sequencing, which is a focus of our initial August launch. So to design this probe, I only need to input two things into the Element custom designer. The first is the flanking sequence surrounding our target, in this case, the spacer region. And the second is the number of bases that you want to sequence. That allows the software to generate an RT primer oligo. That's the primer that I showed on the previous slide that we use to encode and amplify the RNA. It will also generate a sequencing primer within the expected cDNA region, which is used after amplification to initiate sequencing. The user interface will design both of these sequences for you to optimize probe hybridization and minimize wasted cycles. I should also note that as a part of this designer, we'll provide these sequences to you so you have the flexibility to order your own oligos. So overall, we designed this process to be highly flexible, scalable, and fast. Okay, so with the chemistry and design introduced, now I'm going to talk about some of the improvements to data quality that we see in our direct sample sequencing. So one of the major advantages of sequencing of transcribed cDNA is the increased specificity that comes from alignment. So for context, when you hybridize a barcode carrying probe, which is currently the standard in the field, there is increased potential for false positives. And ultimately, that's because there's no way to know if the counts come from a true or a false detection. This manifests as reduced correlation to RNA-seq below some certain abundance, as you can see in the blue points in the graph on the right. Our direct in-sample sequencing overcomes this limitation by utilizing alignment of the transcribed cDNA as an additional specificity filter. That removes any non-specific reads, and you can see the improvement that this provides in the red points on the graph, which maintains correlation down to very low abundance targets. So overall, this results in improved specificity, and you can have very high confidence that every detection that you see is real. So to summarize the advantages of our chemistry and probe design, the single primer binding site removes complex design requirements. Our targeted dual primer design uses knowledge of the expected cDNA to enhance specificity. Our optimized primer placement allows us to sequence further into regions that we care about. And finally, we add another layer of specificity by aligning our cDNA readouts to a reference. So now that I've talked a bit about the chemistry itself, let's go into a little more detail on our 3' transcriptome assay. So in addition to the high specificity that I've talked about in the last few slides, Teton Atlas was also designed with sensitivity in mind. So the fundamental challenge with sensitivity that every imaging-based platform encounters is optical crowding. Essentially, the fundamental limitation of how many spots you can resolve informatically within a finite space of a cell. So as Claudia mentioned previously, both Teton and Teton Atlas overcome the inherent optical limit by separating detections into distinct batches. Every batch has a unique sequencing primer, which means we can sequence different sets of targets in different sequencing rounds. To put this another way, the detections we see in batch 1 are fundamentally distinct from the detections we see in batch 2. And that allows us to increase the sensitivity proportionally to the number of batches that we use. So as an example, in HeLa, we've detected roughly 600 counts per cell per batch, which allows us to get up to 3,000 counts per cell in total across all 5 batches. And if you were to read all 3,000 of those at the same time without batching, that would be a colossal mess, and you wouldn't be able to distinguish anything. So that is how Element gets around this fundamental limitation. For those hoping to get the most out of their batch real estate and really maximize the number of productive detections, we also plan to offer the option to include depletion of undesired, highly expressed targets, stuff like housekeeping genes or ribosomal RNA. This will be an optional and customizable step prior to starting the run, where you can choose the targets you want to deplete. We've seen greater than 90% depletion of the intended targets on average, resulting in greater sensitivity to biologically relevant genes that we actually care about. Okay, so getting into some data validation, I really want to again emphasize that we're reading the actual transcribed RNA sequences in this assay. And as a result, we can do things like sequence alignment, 3 examples of which you can see here. We show accurate detections of SNPs and indels, as well as demonstrate stranded alignment, and we can perform targeted sequencing to regions of interest, such as splice sites. And in order to demonstrate a practical application of this 3' transcriptome assay, we applied direct in-sample sequencing to cell typing. So we cultured A549, HeLa, HepG2, and SH cell types on a flow cell, fixed them, and loaded them onto a VD24 with the Teton Atlas 3' kit. No other off-board preparation needed. At the conclusion of the run, we were able to effectively distinguish between all 4 cell types using only the RNA data. I also want to note that we ran this internally with only 3 batches and no depletion as a test case. So even without the most optimized sensitivity conditions, we're still able to achieve good clustering of all 4 cell types. At launch, we'll be able to enable even more batches, up to 5, to further drive sensitivity. But this is positive proof of concept that if you want to allocate some of those batches to other modules like targeted sequencing, you can still get great data quality from a lower batch allocation. I also want to reiterate again just how simple this process is for the user, in this case, me. We're able to achieve this data with our fully onboard workflow. Just fix the cells, add cell paint probes, load the instrument, and go. There's no library prep, no extra equipment, no cell partitioning. Yeah, I really want to drive home the point of how simple it is to generate great quality data like this. And as great as the RNA data is, that's not the only modality that Teton Atlas will provide. So all of our direct in-sample sequencing methods are going to be natively compatible with both both cell paint and protein. So the low output cartridge launching in August will include 3-plex cell paint, shown here with cell membrane, nucleus, and actin. You'll also have the ability to image up to 88 custom protein markers that can either be provided by the user or mix and match with up to 3 pre-designed and validated focus panels. In the Teton Atlas high output cartridge that's coming in Q4 of this year, we're going to add the capability to include 3 additional cell paint markers. Okay, so now that I've shown the details of the technology, let's take a deeper dive into a specific use case that this enables, specifically optical pooled screens. So as a very brief introduction, optical pooled screening is a method of pooling CRISPR knockout libraries together for highly multiplex screening of genotype-phenotype correlation. This is a fairly new technology that is valuable in biomarker discovery, interrogating mechanisms of action, many other things. So how is OPS done today? Well, most people are doing this by hand or on custom homebrew equipment setup. And it's typically a long process that takes greater than 1 week for a screen. Additionally, these screens typically rely on a single omic readout, typically around a dozen cell paint targets, in order to assess the biological impact. So there are a few challenges here for the field today. The first is throughput and automation, and the second is increasing the omic capability for richer information content. And Element can help with both of those. So in terms of guide RNA detection, people have been trying to automate this sequencing readout on sequencing platforms for some time, but it's quite challenging. So the visualization of single guides is complicated to do with barcodes or with padlock-based approaches, as you need several unique probes for each spacer. That makes it very difficult to scale. As an example, if you wanted to do a whole genome screen, that would require tens of thousands of unique probes. But with our direct in-sample sequencing, you only need 2 primers, and you can look at any number of spacers all the way up to whole genome screens. That's because we have true sequencing as the readout of the spacer RNA, not just a barcode. Here are 2 examples of cell types that we have sequenced guides in. On the left are smaller MCF7 cells that form tight clusters and grow colonies of cells with the same perturbation. And U2OS cells on the right have a more classical epithelial morphology, where you can clearly see a single guide sequence within each cell. Using direct in-sample sequencing, we have a good rate of success for aligning cells to these spacer sequences, which is the ultimate readout that we want from this assay. That brings me to an early demonstration of direct in-sample sequencing for optical pooled screens. This was a collaboration with the lab at Genentech. And I'll also note that this story was recently presented as a poster at AACR. And I would invite everyone who's interested to take a look at that poster by scanning the QR code here. In this experiment, we tested our platform's capabilities for pooled screening by using a modified workflow where we combined the Teton protein and RNA fixed panels with guide RNA sequencing. Most optical pooled screens typically use only cell paint as a phenotypic readout. So our goal here was to produce a much more rich data set incorporating high-plex multi-omic content. This was a 500 gene perturbation study. And to each of those 500 gene knockouts, we assigned about 2,000 cells, which is more than sufficient for statistical analysis. And in our Teton panels for RNA protein, we generated about 1,000 counts per cell from the 400 targets present in those panels. And within this data set, within this experiment, the first thing that we set out to do was validate our assay against expected biology. So our cell paint includes calreticulin as a marker for the endoplasmic reticulum. So when we looked at the perturbations that cause the most regulation of our cell paint ER signal, we were happy to see calreticulin knockout as producing the most downregulation of any knockout. Similarly, we saw that the reduction in mitochondrial cell paint signal correlated well with expected biology, as the 5 perturbations that caused the most downregulation of this signal were all mitochondrial ribosomal proteins. So I think this was a nice validation of our cell paint as well as our ability to call these guides. We can also look at individual examples of expected biology with other omics like RNA. So for example, a CRISPR knockout of the ribosomal protein L8 induced dramatic upregulation of cyclin-dependent kinase inhibitor 1A and downregulation of ATPase family AAA domain protein. This is represented in the far right panel, where cells expressing RPL8 guide RNA showed an increase in blue spots and a reduction in pink spots. These guides are all implicated in cell growth and cell cycle regulation, so them being related in this way conforms well to the expected biology. So the prior slides have showed a few specific examples, but there is a lot more data here with over 1,000 multi-omic features segmented by over 500 gene knockouts from the CRISPR library. So one way we can look at the data is to observe multi-omic changes in RNA, protein, and cell paint features for any given gene knockout. Following up on the last slide, we can look at the regulation of all features as a result of the ribosomal protein L8 knockout. So in addition to the 2 genes that I mentioned on the prior slide, there are many multi-omic features here that were altered as a result of this knockout, not just those 2 and not just the expected results. So with a traditional single omic cell paint readout, which again is the most common type of optical cold screening done today, a lot of this detail would have been missed. And to really drive that point home, here we can compare the UMAP clustering of gene knockouts with cell paint only versus with the full multi-omic readout of cell paint RNA and protein. So in this experiment, when we look at all of the multi-omics together, that significantly improved our ability to cluster phenotypes versus cell paint alone. And I would say the takeaway from this experiment is that we were able to correlate phenotypic and transcriptomic changes with the CRISPR genomic perturbations in a way that's never been possible before. This is really the first time that an optical pooled screening data set this rich in multi-omic features has been generated. And with that, I'll conclude the presentation of our scientific story collaboration with Genentech. If you're curious about more of the details, again, I highly recommend downloading the poster from AECR and giving it a read. And I'll wrap up by highlighting the application of Teton Atlas to optical pooled screening, which is really a featured part of our August launch of the low output kit. And I'm so excited to see what the community is going to do with this technology. And on that note, I will pass the mic back to Jens.
All right. Thank you very much, Claudia and Dan, for that exciting presentation. Before we move on to the Q&A section of this webinar, I do have 2 very exciting updates to announce. Number 1 is the slide that you see here. I am excited that the Teton Atlas low output kits and the direct in-sample sequencing technology is now open for pre-orders today. Please scan the QR code on the left on this slide for additional information on how to get started. Please contact also your local Element support team to learn how you can get your hands on this early and plan for your experiments. In addition to opening Teton Atlas pre-orders, we're also excited to kick off a global grant program and are now accepting applications for the Teton Atlas. This grant program will support your multi-omics research on the AVD24 platform. The winner will receive 1 Teton Atlas low output run, which will include 3 cell paint markers, targeted RNA sequencing up to 100 cycles, and then up to 88 custom proteins of your choice. One of the key applications that you've seen in this presentation is the ability to combine morphology, proteomic readouts, as well as targeted guide sequences or any barcodes, all from the same cell at scale. But I'm sure you can think of any additional applications, and we'd love to hear from you on where you want to take this assay in your research. Samples will be run at Element and receive full project support from our applications team. Applications are open today and will close June 20th. For any additional information, please scan the QR code to your right.
尼尔生物公司是专业的生物技术服务公司，提供空间转录组服务和 Element 组织原位单细胞检测服务，还提供单细胞测序服务和各种基因测序服务。欢迎惠顾。